This meeting is now being recorded.
Thank you for joining us for Metabolon's half-hour webinar series, where our topic today is Precision Metabolomics, Quality Control and Assurance in Untargeted Metabolomics. We will have a short period of time for Q&A at the end of this webinar. Please feel free to submit your question via the Q&A section at the upper right of your screen. And just a quick reminder, the webinar is being recorded. Thank you. Your presenter for today is Dr. Ann Evans. Dr. Evans leads the Global Metabolomics and Lipidomics Profiling Research and Development Team at Metabolon, where she has been working for 13 years. The metabolomics and lipidomics platforms developed under Dr. Evans have been the analytical basis for thousands of commercial studies from over 500 clients since 2004. She has publications covering the analytical methodology as well as the informatics and data processing requirements for global profiling workflows. Dr. Evans received her PhD in chemistry from the University of Virginia, where her research was focused on biological mass spectrometric detection of protein and peptides with renowned professor Dr. Donald Hunt. Currently, much of her research is focused on developing the global metabolomics and lipidomics technologies, as well as new technologies, to be able to support initiatives into large human cohort studies to study human health while also making the technology precise and robust enough to study individual patient health, precision medicine. I'll be turning the presentation over to Dr. Evans now.
Good morning, and just to reiterate, thank you everybody for joining us today. I'd like to start off with a brief apology. I've got a bit of a head cold today, so all apologies for any sneezes or coughs that I have during this webinar. I got my coffee here, so hopefully we'll be able to keep going without too many problems. As Brandon introduced, I'm going to be giving a talk today about the various quality control and quality assurance practices for our untargeted metabolomics methodology that we use here at Metabolon. Just as a brief overview, I'm going to go over a little bit of what is metabolomics, why do we like metabolomics, and how do we do the brand of metabolomics that we do here, and then leap off from that into Metabolon's quality management system, including, some of our QA and QC practices, which of these are just sort of good generic metabolomic practices. And then I also want to conclude the talk with a little bit of discussion about how we then control for samples that are run under this relative quantitation methodology when you have something that's going to run for many days or even months and potentially years in some of these larger cohort studies. So first is the what and the why and the how. So metabolomics is the survey of the small molecules in the sample, and one of the reasons that we really like looking at metabolomics is because it's really the end readout of a number of different functions, right? So A, you get the downstream effect of your normal endogenous processes. You have your genes which control for specific proteins in the body, and then those proteins function by way of using these metabolites to carry out various functions in the cell. And so by monitoring these metabolites, these endogenous metabolites, if you will, you get a good sense of what is going on within the individual with their own endogenous processes. But in addition to that, when you study metabolomics or the small molecules present in these samples, you're also going to get a really strong sense of what are the other influences which could be impacting the health of that individual. So think of things like the microbiome. The influence of the state of our gut health, or any particular study's gut health, has a profound impact on how we're able to function, whether our gut is doing a good job maintaining that barrier, whether you have some drug tox, which is leading to more of a leaky gut syndrome, and certainly if you're talking about things like inflammatory, IBD, or different autoimmune diseases, that can have a huge role. But understanding the microbiome and the influence it has on its biology on affecting our health. But then in addition to that, you also have this entire side of it where you're able to see so much of the individual lifestyle and environment of the organism, whether you're talking about a free-range human, that pops 2 Tylenol before giving plasma that day, or whether they exercise or not, or happen to eat a lot of ginger in their diet. These are all things that have signatures within the metabolomic space. And so we really like to think of metabolomics as being much more than just a sort of an ability to look at the endogenous, risk of a disease, but really much closer to the idea of it being the status of what is currently going on in the organism, and that you can see this much wider range of influences on the health of the organism. So what we're going to do at Metabolon and within Metabolomics is to, as much as we can, cover as much of that biochemistry and biological pathways as possible, so that in any particular disease, you have molecules which you can map to all these different areas of key metabolic pathways. Now, this just happens to be the map for sort of known endogenous chemistry. If we had bacteria metabolism, there would be a separate wing of metabolism, and drug metabolism would have its own set. This is really just the endogenous chemical map. But at the end of the day, we really want to be able to have markers in every range of biochemistry so that in any particular perturbation or drug treatment or whatever it may be, you can see where that biological insight is or where those biomarkers are showing up in that particular study. So this is not a small challenge in that there is a diverse range of molecules that are really covered. When I say diverse range, I mean the chemical properties of all of those molecules are really quite extreme. You have very polar metabolites that are these carbohydrates, which need to be analyzed in a very different way than, say, very complex lipids, which are the lipids that make up all of the membranes in the body and which are responsible for energy transport within the body. And unfortunately, there's really not one technology that allows us to be able to detect all of this breadth of chemical diversity. And so what we have to do within the metabolomics space is really run multiple methods. And this is actually one of the first aspects of a quality system, is that the fact that each of the methods that we run is really individually optimized for specific classes of compounds. And this is one of the ways that we can assure that we have a really strong underpinning of data for every type of compound we want to analyze. The other thing that having these multiple methods does is it gives us actually redundant measurements of the same compound. So something like 70% of the molecules that we're able to identify are seen on one of these multiple platforms up here. And so that means if I see leucine, and leucine seems to be particularly important in my study, I can also look to see, well, I also detected leucine on this NEG method. Do the results from those match? Are they correlative? And that is a nice internal quality control check with this data. So having these multiple methods not only gives you greater coverage and greater scope of metabolites that you can detect, again, reaching out towards that, getting that coverage so that you can see where the metabolic signature may be, but also gives you this nice internal quality control checks when you can see the same molecules detected on multiple platforms. So I'm going to focus in very much today on just the metabolomics workflow. We have a separate complex lipid methodology, but this has a very different set of quality control, quality assurance systems in place. And so I really want to just focus our attention on the metabolomics workflow, which has some more specific quality control measures for that technology. But again, I just wanted to let you know that we do have a separate method for the complex lipids, but I'm going to be focusing my attention on the metabolomics platform. So we do run these 4 separate methods, and I'm not going to go into them today because it's really not the focus of what I want to be able to talk about. So the methodology that we ultimately use, or I should say that our informatics workflow from the ground up is really designed around optimization for quality and accuracy. So when you think about the traditional untargeted metabolomics workflow, typically what you see is that people will collect their data and they will generate a feature list, or basically a peak list, a list of all of the peaks that they found in their sample. They will attempt to do some sort of an alignment across the entire study so that there's some sense of this peak is the same as this peak across samples. And then they take all of that data and they attempt to make sense of it. The problem with this sort of traditional metabolomics workflow is really that it is absolutely fraught with a lot of potential errors. People make assumptions about the identities of these features and then end up making a lot of misidentifications. There's also potential for a lot of misalignment of data so that your accuracy of quantitation across this type of analysis is also quite difficult to verify. So Metabolon really from the very beginning said, look, we've got to find a way of deconvoluting this feature list, because what we know from a lot of data is that a lot of these features are simply redundant measurements of the exact same compounds. And there's some really great publications out there. And if you want to send out an email, I can send out some of those publications if you want to see the ones I'm talking about specifically, which really start delving into just how much of these peaks that we're detecting are really just either noise or redundant measurements of the same compound. So when you work in this workflow where you're trying to pay attention and identify all of these individual features, you're simply going to have too many inaccuracies. So Metabolon's workflow is really designed to take this very large list, of which, again, we also detect hundreds of thousands of peaks when we run a sample, and quickly as possible get it to a highly curated list of just those compounds which we are detecting in those samples. And it is this process of taking all of these really complex, very raw, unfiltered data and moving it to this highly curated list, which is where many of our quality control and quality assurance processes are placed. We want to ultimately get from that incredibly raw data to this curated list such that after you have this very curated, highly accurate list, only then do you take those compounds into some sort of statistical analysis and then move that into interpretation. So a lot of what I'm going to be talking about today are all of the processes that we have in place in moving this data along this pipeline to get to this list of identified molecules that are really highly accurate and both in terms of identification but also in terms of quantification. So one of the first things that we do, and I mentioned this here, that this important filter here, going from the features to the identified molecules, really one of the first steps in that process is making the identifications of the molecules that we are seeing. And we do this through use of an authentic standard library. So what we've done is that we have purchased at this point over 4,000 commercially available standards. We've also done a lot of synthetic work of molecules that we are confident that we have in the sample, but we couldn't buy them commercially, and so we've synthesized them so that we can track those. We take all of those standards and we run them on the analytical platform, and we document the exact characteristics of that authentic standard on the platform. So we'll document the retention time of that analyte. We'll document the mass of that analyte, including all of those ions and additional features that it produces. And then in addition to that, we also collect all of the fragmentation spectrum. So that means when I detect an analytical signature, in addition to all of those redundant measurements from the same compound, I can immediately identify them all with a very high accuracy because I can say, this signature that I'm seeing has the correct retention time for leucine, as an example. This has the correct accurate mass for leucine, and I can see the adduct of that leucine. I can see the in-source fragment, and I can further confirm it with the fragmentation spectrum for leucine, and again, give a very high confidence call. So this is called a Tier 1 or a Level 1 confidence identification as specified by the Metabolomics Working Group. And this is really one of our key first areas of quality assurance. You're not going to be able to do good metabolomics, untargeted metabolomics work if the identity of the molecules that you are providing is at all in question. Right, so this is a really key part of the accuracy of the identification is, again, this multiple orthogonal criteria, including rich retention time, which gives you a lot of power to differentiate things like isomers. You know, isoleucine and leucine are very hard to distinguish, but they can absolutely be distinguished by retention time. And this phenomenon is extremely common within the metabolic space. So while we do this workflow, we actually monitor this process continually, right? And certainly quality control or any quality management system is really about monitoring the process and seeing exactly what's happening. And this plot along the bottom that I'm showing here is actually a distribution of RSDs for all complex or for plasma samples, for all the compounds that were detected in plasma samples and shown by year. And what you can see is that back in 2005, we were not doing so great, right? In a technical matrix or in a technical replicate of human plasma, we had an average RSD of 48%, and we were only detecting about, 100 compounds. But over the years, we've monitored this process. We've put various hardware as well as quality control processes in place, such that as of 2016, you can see we've got a very nice distribution where the average CV for the number of molecules we're detecting is less than 5%, and we can do that with detecting and identifying between 2,000 and 2,500 molecules. And the nice thing about this tracking process is that at any point I can still go in and interrogate exactly what molecule is it or what classes of molecules that are still giving me this higher process variability, and then we can make improvements to actually be able to shift or change things as we can to try to shift that distribution ever and ever lower. So some of the things that we've put in place over the years with that goal in mind are really, some of these are not the most exciting things, but again, it's a part of a quality management system. So things like SOPs, fully documented and maintained SOPs that are reviewed by multiple people and staff, fully standardized extraction procedures and analysis procedures which have to be followed and checked, calibration reports, a lot of documentation. All of our systems, samples, data, everything is housed through a LIMS system so that I can go with full traceability. I can go back to a study I ran 10 years ago and pull up the peak that we used to call leucine in the particular disease state. So it's all very fully traceable. We use very advanced technology, and that absolutely improves the quality of the data. And even something simple like Proficiency trained staff. All of our staff are trained every 6 months and have to demonstrate the ability to effectively run metabolomic studies, and there's various quality control metrics that we can use to be able to assess that they're doing their job well. In addition, we have, things like just infrastructure controls, that the lab temperature is monitored and within criteria, that the freezers are always maintained and at appropriate temperatures so that we can immediately be notified if a freezer is starting to go out of specification. And then we've already talked a little bit about how our entire workflow is actually really optimized around quality and accuracy, and that we do take this, what our opinion is just too raw data that is this feature data, and we really curate it down to the list of really accurate compounds and quantifiable species. So some of this I'm going to talk about in more detail now is sort of what are the visual quality control steps, what are the approval processes for our automated systems. So I'm just going to start right off into some of the specific examples of the things that we do with our untargeted metabolomics. So as with any quality management system, there are a number of different strategies that we use. One of them is really traditional quality control standards. These are isotopically labeled compounds that are spiked into every sample we run. These come in two different flavors, if you will. Recovery standards are spiked in at the point of the extraction so that we can make sure the extraction goes reproducibly. And then we also have separate internal standards which are spiked in just before the instrument analysis. Now these specific standards are really used to allow us to really triage, if you will, how well the samples ran the night before. So we do things like we track those internal standards over the course of the night's runs. You can see these red bars here are our quality control limits, so that we know exactly whether or not the data that we generated the previous night meets our quality control specifications. So we check things like mass accuracy. Did the instrument drift in sensitivity overnight? Did the instrument drift in sensitivity? And as well, you can see the red bars here showing what is acceptable and what is not acceptable. We check consistency of fragmentation. We know that, even though you tune and calibrate an instrument to produce a consistent fragmentation spectrum, how consistent is that? How consistent is that tune? So we always maintain a check to make sure fragmentation consistency is high so that it enables the library match more effectively. We also have chromatographic performance checks where we look at peak width at half height as well as looking for separation of critical pairs. And we have these for each arm of the platform so that we can be able to monitor really the health of the column to know if we're on an edge or that the column needs to be replaced before we run the next study. Very much similar to a sort of a system suitability check where you know that before running that night's runs, your columns are near the death point and so you need to replace them. At the end of every study, we actually have about a 15 to 20 page report which is generated, which goes through all of these things that I just mentioned per analytical method. So it ends up actually being a 75 to 80 page report that we go through and review all of these things, again, mostly looking at those internal standards, but also looking at a few endogenous molecules like the critical pairs I was talking to you about to really be able to vet that we are happy with how the instrument ran the previous night. Now, the other thing, in addition to these sort of traditional QC standards, we also have entire samples dedicated to quality control, and these also come in multiple flavors. We have pooled technical replicates that we run. Ideally, depending on the matrix, we want the technical replicates to be the exact matrix of whatever the client study happens to be. So if you're running monkey brain, we would like to have a technical replicate of monkey brain throughout the entire run. And these technical replicates actually serve a couple of really important points in terms of monitoring the data. I showed you this chart earlier. This chart of being able to watch our precision were generated from those technical replicates of those biological samples, right? So because we always run these technical replicates, we can always be able to monitor the process and know, is the run that we collected last night, are the same number of compounds detected as we normally see, and are they detected with the same amount of precision as we normally see? And that gives us a really good sense of whether the platform is operating under control. And I'm going to talk to you in a few slides about some of the other uses we can use for that technical replicate matrix. We also have a water blank. This is really an ability for us to be able to identify and really be able to remove those compounds that are introduced to the sample because they've been stored in plastic tubes. You know, every sample comes to us has been in some sort of plastic tube, and those plastic tubes have molecules in them that get leached into your sample. We want to make sure we know exactly what those compounds that are coming as a result of storage and handling, but really don't have anything to do with the biological samples, so that we can really focus in on our attention on what's the actual biological signal. And then finally, one of the things that we'll do is proficiency samples, right? So these are, depending on the level of quality and the level of rigor with which your samples are having to be run at. If you're running clinical samples, then you have to run precision sample or proficiency samples every time you do a clinical analysis. For more of a traditional non-targeted RUO type metabolomics studies, you can do proficiency samples every 6 months or yearly. So really it depends on the rigor with which the data is going to be used on the back end. Proficiency samples are generally just samples where you know there is a signature you're looking for, and then you can track it to know that, yes, these samples gave me the same results today as they did last week, as they did 2 months ago, as they did 2 years ago. So now I want to start talking about a slightly different aspect of what we do. We talked about sort of really the traditional quality control steps, but a lot of this is quality assurance, just the processes that you have in place to be able to really look at the data post-collection to be able to assess whether everything that had to happen happened well and that it is ultimately the best representation of the data. So we already talked about this aspect of it, that all of the identifications that we are making are going to be these tier 1 identifications, right? The highest level of confidence identification, and that's a really key point to being able to, have confidence in your identifications. But in addition to that, because we're working on our way towards that curated list of metabolites, what this workflow allows us to do is actually hand and manually review every compound call that we want to be able to report out. So what we're going to do is we're going to pull the data and we're going to look at it. And this is what is the current proposed, say library call for 1,5-anhydroglucatol in this particular study. We're going to go in and look that we like that the quantitation of the peaks or the integration of the peaks was done well. We're going to go in and look that this sample or that this compound was not seen in those water blanks so that we know this is not something that we've contributed to the samples. And we're going to also manually vet that the quality of the fragmentation spectrum match was a high enough quality to the library to give us confidence that this is a correct call for this molecule. We're going to check that the same peak was aligned across the entire data set. Now, part of the reason we're able to invest the time to do this is because we're not trying to do this for every single peak that we detected in the original data. The library has really allowed us to get to a much smaller list of potential compounds, and then we can really spend the time at making sure the calls and the integration of these peaks are of the highest quality. The other thing that we have done is as we go in and really start looking at where are the sources of variability in our samples and what is leading to have, again, some of these molecules that still have higher variability than maybe we like, we saw that a lot of it was related to the consistency of these automated software programs, right? We all have to rely on automated software for our peak detection and our peak integration, as well as a lot of us for the library identifications. Now that is a lot of responsibility on an algorithm, right? And any algorithm is going to have weaknesses and flaws. So when we started looking at molecules that had higher variability, we noticed that in a lot of cases it was related to peaks like this, where you had two compounds that were partially resolved. This happens to be an example of two different lysolipids. One's the one form of the lysolipids and one is the two form of the lysolipid. In most of the cases, the software was able to see on a sample-by-sample basis that there were these two peaks there and able to integrate them separately. However, the algorithm, our automated software, in two cases was unable to distinguish that front peak, and so unfortunately what you had is two negative effects. One, the second peak now looks quantitatively larger than it should be because it summed the area for that first compound in, and you have also now lost detection in that sample of that front peak, so you have a missing value. So we wanted to go, and I should, one more example here is that it actually drew a tangent instead of doing a baseline drop for, whatever reason. And when we went in and saw this, we said, well, this is something that we can actually quality control for, right? So we developed software that went in and actually goes through and looks for these inconsistencies in integration across an entire study, and then we'll say, look, I recommend, based on majority rules, doing consistent integration by doing a baseline drop, and a baseline drop. Now, you can argue whether a baseline drop is the best way to integrate those peaks, but we want to make sure whatever we're doing it completely consistency to be able to give the most accuracy of quantitation. Just putting this bit of quality control around how peaks are integrated within the study dropped our process variability 25%. So that was a significant amount of process variability being introduced by these automated programs. Now finally, the last thing I really want to be able to talk to you about today, and it's certainly always something on the minds of people doing these much larger metabolomic studies, is how do we start correcting for variation in the data which is not coming from biological variation. We know, running mass spectrometers over days or months, that there's inherent differences that come in the sensitivity on any given day, and that, if left uncontrolled for, can actually mask quite a bit of biological signal. So we have a couple of different strategies that we use that we like a lot, and I want to talk about both of them. They both have advantages and disadvantages. The one we like the most is what we call block correction, and block in this case we're defining as a day. So if you think about this as each one of these days is the spread of values for a given compound. So just so I can pick on a specific column, let's say this is the spread of intensities of an alanine on day 1. This is the spread of values for alanine on day 2, day 3, day 4. And you do this on a per compound basis. That's a really important part of this. What you do is then you divide all of the values for alanine by the median on day and essentially then register everything to a value of 1, thereby correcting for those day-to-day variations. And this works incredibly well, and we've done a lot of research testing this against targeted assay data to be able to show that this is a really good way to do it. But the key assumption here is that this comes with an extremely well-balanced study run order. If you have more females run on day 1 than day 2, you're going to normalize out that difference. If you've got more case and more controls on any given day in a study, that can absolutely hurt the ability of this normalization, and you will decrease the accuracy of the normalization. So there are advantages and disadvantages. An advantage is that you don't have to have any additional samples. You are just using the data, the samples that you have in the study to generate this normalization. So again, this works extremely well. This is our preferred method, but it does have some limitations in terms of knowing ahead of time how to appropriately balance the study. The other approach that we've seen works very, very well is the use of either a normalizing sample or a bridging sample. This is where you run the same set of samples, ideally a number of replicates of it, and then you run those same set of normalizing samples over the course of the entire study. So in that way, you always compare the experimental samples back to that normalizing or bridging matrix to be able to do that similar correction, right? This takes away some of the weight of having to have such a perfect balance study. But again, the key limitation here is you have to have enough of this normalizing sample to really support running that over the breadth of the entire study. It also comes with the assumption that that normalizing matrix is incredibly stable. If you're starting to have that normalizing matrix break down over time, that's going to lead to some issues. So these are two really nice strategies to be able to account for that over time analysis in terms of the relative quantitation. Both of them do very well when you compare them to targeted values, and we've seen very good demonstrations of those working. So just in summary, I know we're about a minute over here. I just want to say again that there's quite a few quality management processes in place, all the way from sort of the mundane things of having SOPs and proficiency trained staff, full traceability and tracking of data and archiving of data, all the way through the entire workflow really being optimized to allow us to do what we do so that we can have the assurance of being able to feel confident that not only the identifications of the molecules we're providing, but also the quantitation of those molecules are accurate. And then some of the strategies that we've used when we normalize samples that we need to run over larger periods of time. So thank you very much for joining today. I'm going to let my colleague take over for a minute here.
Thank you. We'll be answering questions now in the order they've been received. Since we're a little bit over on time, when we run out of time, we will answer your question offline as soon as possible.
So I'm just going to read some questions off here. When studying many genotypes, for example, over 300, the diversity comparison or GWAS, how do you handle missing data or data normalization? So missing data is obviously something that, all the different omics technologies, has to find ways of handling. We generally go with the assumption that missing data is a result of limit of detection of the instrument. We do not generally have data that is missing just as a result of, it's a big signal and it wasn't seen. Most of our missing data is as a result of limit of detection of the instrument. So we impute values for our missing values at the lowest value seen from the experimentals. And then that actually enables, a limitation of the skew and statistics that can happen if you do not deal with the missing data. So hopefully that answers that question. Are water blanks included with each run and each specimen? Yeah, so water blanks are an absolutely standard part of every analysis. So we always run 2 in every set that we run. So that you can also use this for tracking background contamination within solvents, and there's a lot of good things that can come from just running a water blank, so we run them in every study. If certain peak position or peaks have several corresponding compounds, how do you know which one is the right one? Again, this is a really good question. So one of the key things that happens, again, in metabolomics is that there are many isomers, right? All you have to do is think about, like at the hydroxybutirates, you have 2-hydroxybutyrate, 3-hydroxybutyrate, 3-hydroxyisobutyrate, 2-hydroxyisobutyrate. These all have exactly the same mass and exactly the same fragmentation pattern. The only way you can distinguish which one is which is through retention time. Now, so by running the internal standards, we know exactly the order with which they are received. Now, where it can be tricky is, let's just say in a particular study, you only see 2 of the 4 peaks that you might expect. And in that case, you're going to be relying on historic precedents of which are the more common ones, which are the less common ones. In some cases, if we are not completely confident that we are making the right identification, we will actually not report that compound. Again, we are fairly conservative in that we want to make sure that when we identify a compound, we make it correct. So hopefully that got some of that question. How many specimens are typically included in each run? Is the number of run days included in the output provided? Yes, so you will, in the data that's provided, we'll actually show you the sort of which run day was run when, at least I believe that's part of the data delivery. How many specimens? So individual samples in a set can range. We run anything from 50 samples in a study all the way up to many, many thousands in a study. What we consider a batch is 48 samples, and then we run 4 batches per night so that you get a sense that we can run about 140 of these experimental samples a night, and we do about 180 total injections per night when you add up all the quality control when you run all the quality control samples in there. The only other thing I wanted to mention about that is we do, within a project, you want to maintain that it's the same type of sample, right? So serum, if you're going to be running a serum study, make sure all of your samples are serum. Don't mix EDTA plasma and serum. That's going to certainly cause issues within the data. Let's see, I've got a couple more questions as long as we're okay on time. Okay. So one other question is, can you comment on the use of preclinical species? So certainly, a lot of our research, we span really all sorts of levels of whether we're working in the preclinical space all the way up to working in the clinical space. A lot of the sort of practices that we talked about today are really for our more, everyday RUO preclinical work, right? So you don't necessarily have the rigor you don't have to have the rigor of regulations that you would have if you were working in the clinical space. We do support all the way up to the clinical space, but certainly we do a lot of work within mouse models, cell line cultures as well, all using these same quality control metrics and adapt them where we need to. Interpretability and submission guidelines on publication. So certainly we are very well aware of all the publications that are out there that are adding more and more criteria for the principal investigators about what things they need to be able to provide. We are actively working with each of our clients to really understand what they're hearing from the journals, and we're actively working on different ways of satisfying the journal's requirements. So certainly some of the things that we're discussing is being able to provide all of the quality control data with all of the clients so that they can use that as a mean of publishing that along with their experimental data to show the quality of the platform. And we're also in some early phase discussions to be able to provide the raw data where some of the high-end tier journals are now requiring that you have access to the actual raw data as well. So we're in early discussions with figuring out how we can manage to provide that data as well to check off some of those publication requirements. Can a general statement be made about samples for analysis and the amount of sample requirements? So we have run just about every type of sample analysis out there, or every type of sample matrix you can imagine, all the way from, certainly plasma, urine, feces, cell culture, tissue culture, biopsies of tissue. We've even done, one of my most favorite ones are the dental scrapings from 200-year-old mummies. So, we didn't see much in the dental scrapings of 200-year-old mummies, but we did actually attempt to run them. We also do a lot of consumer good products, so things like yogurt, chocolate, orange juice, people who are just trying to understand some of the biochemical profiles of again, yogurt, particularly in this world of demonstrating that your yogurt is better than somebody else's yogurt in terms of establishing biome. Sample requirements, we generally request about 100 microliters to 150 microliters of plasma, and a similar sort of amount for cell block, and then tissue amounts depend a little bit on density of tissue and whether it's a dry weight or a wet weight. So that's a general sort of description there. And the last question I think I'll answer here is the question specifically around research in bacterial studies. Certainly we do a lot of work in bacteria. Certainly the microbiome aspect of this is humongous. One of the things that's really excited about metabolomics is that we can see so many of the influences of the microbiome and bacterial metabolism, and it's in fact that it has, because they have unique genes and unique enzymes that can produce unique novel metabolites that are not seen in human species. So we do a tremendous amount of work in this space, and it's a very exciting place to be right now because of all of the research coming out showing just how important that is. So again, I think that's all the time. I ran about 10 minutes over. I appreciate your patience for staying on the line today. Again, if you have any questions that we didn't get to, you can feel free to add some more questions here, and we'll answer them online afterward. Thank you very much.
Thank you for attending our webinar. In case you'd like to share the replay or listen again, the recording will be available soon on our website at www.metabolon.com.